Now we are moving on to objective 2, because now we have developed the methodology. We've shown that we can induce it in E2 in the lab in Xenopus. So now we're going to expose the Xenopus through metamorphosis to the water from selected watersheds to see if we see estrogenic effects there. Here is our sites. Here is where we had Snow Creek, and this is Cane Creek and Chocolate Creek, which are the 3 sites that we selected, and I'll discuss them a little bit more in a minute. Here you can see a zoomed in map of Snow Creek. Right here is the railroad trellis, or not railroad trellis, there's a railroad track running along the site. You can also see industrial Aniston all around it. Here is Cane Creek. It was south of a local dairy farm. Here is the farm, and here is the site along Highway 431. And this is Choccolocco Creek out in Rabbittown, actually. Here is Brown Bridge Road, and this is where our site was. You can notice that this is just strictly an agricultural and rural area. Now, Choccolocco Creek was immediately downstream of Talladega National Forest. It had its potential non-point sources were very minimal. It only had 2 bordering residences and a pasture. This was actually what we used as our reference site. Cane Creek is south of a local organic dairy farm, and you saw that there. Its potential non-estrogenic sources were stormwater runoff from Alabama Highway 431, effluents from Fort McClellan, and airport drainage. Snow Creek is a well-known EPA Superfund site for PCB contamination. Potential non-point sources for it include PCBs, industrial effluents from downtown Aniston, and then once again stormwater outfall, as this is actually where we were collecting, and you can see the stormwater outfall here. Now, for the Xenopus that we exposed in this experiment, we took stage 48 larvae and placed them in 10 gallons of Plastiquaria and 20 liters of undiluted solution. They were maintained on a 16-hour light dark cycle, and temperature was maintained at 19.5 degrees Celsius. You can actually see our temperature regulation system here. This is the reservoir that water is being pumped from, and it's attached chiller. The water is being chilled here. It goes through these tubes here, and then heated chamber coils are placed in each aquarium, and then it recirculates through it, and it's never actually exposed to, or it's never actually in contact with water because it's in the tube. Exposure water. Now for just animal care, the tadpoles were fed 0.5 to 1 milliliter of strained green peas, and newly metamorphed frogs were fed approximately 30 milligrams of 1/16-inch frog gill obtained from Xenopus 1 per frog. We perform water changes weekly. We used AquaCheck 5-in-1 water quality test strips to test water parameters. Water quality parameters included pH, alkalinity, total hardness, and free chlorine. Temperature was recorded daily, and as individuals reached stage 66, they were collected for blood collection. Our treatments included undiluted water from each of the 3 sites that we just discussed, and a negative control of dechlorinated tap water. They were performed in replicate. Each replicate had 20 tadpoles per replicate. The exposure was maintained until 50% of the negative controls had metamorphosed. From previous experiments just raising Xenopus, I found that that's what I could pretty much guarantee within a reasonable time period to actually get the tadpoles to metamorphosis in these tanks. And this actually took 129 days. The larvae were staged according to YouTube and Faber, which is the standard for Xenopus latest development. And blood was collected from a minimum of 5 stage 66 individuals, as that's what, from reading in the literature, we found was the standard number for testing. When it was not possible to do this, we did take blood from lower stages. Now, for the water collections themselves, we collected water in the creek in 5-gallon pails. Here you can see the pails, and here you can see the approximate level or approximate location within the creek that we're actually collecting water from. Before actually collecting water, the pails were rinsed 3 times in creek water. We recorded water temperature and site observations. And before adding it to the aquarium and doing the water change, we allowed the water both the exposure water and control water to acclimate to room temperature for 24 to 48 hours so that we reduce temperature shock to the market. For the statistical analysis, the comparisons were performed with both treatment and age class. The age classes were stage 51 through 65 and stage 66. These age classes were chosen because they bracket vitellogen and gene expression reported in the literature for Xenopus. Covariates were replicate, stage length, and wet weight. Now we're going to actually start into objective 2A because all the things that we've just discussed also apply to the native endurance that we collected. I'm sorry. The results for this experiment was that we did obtain stages for vitellogen and detection in the larvae from this. The vitellogen was detected only in stage 66 individuals, as these were the most common tested, and there were significant mortalities in the control in Cane Creek. Here you can see the developmental stages that they actually obtained, unless otherwise stated. P here is less than 0.05. Letters designate treatments with no statistical differences, and what this means is that you'll note that Cane Creek is the only one with the letter A, so it was statistically different, or statistically lower from all other individuals. Here, the negative control and Snow Creek share a B, so they group together. And Snow Creek and Chocolaca Creek share a C, so they group together. But Chocolaca Creek was different from the negative control. Vitellogenin was detected in the plasma of Xenopus exposed to water from all three sites. The average molecular weight that we saw in this was 197 kilodaltons, and there were no significant differences between treatments. And you can see the graph here for the percent of Xenopus exposed to water from selected sites with detected vitellogenin induction. Although once again we did not see statistically significant differences, you can note that we did do a student t-test for comparisons as to whether or not they were significantly different from zero, as we do not actually expect vitellogenin to be induced in these. And the Cane Creek and the control were not statistically significant from zero, but Snow Creek and Chocolaca Creek were. Conclusions for this experiment, we did conclude that vitellogenin was induced in Xenopus laevis exposed to water from the three initially selected sites. One of the reasons that we may not have seen it necessarily, well, one of the reasons at Cane Creek that it I'm sorry, I'm very close. Okay, vitellogenin was induced in a very low percent of individuals exposed to Cane Creek. The Cane Creek site was downstream of most of the major potential sources that we were expecting. Furthermore, the cattle at the farm were restrained from actually reaching the creek at this site. And another factor that may have contributed to the lower numbers is that in Xenopus, although you can detect vitellogenin at stage 58, the liver hepatocytes are not reported or have been shown to be competent for vitellogenin production in only approximately 1.2% of their hepatocyte populations at stage 62. This can be increased by the addition of triiodothyronine or T3, which actually increases their levels along with estrogen to be 20% competent in producing vitellogenin. The embryos at Cane Creek on an average only reach stages 61, and so may not have been at level or at stages high enough to induce phytogen at high enough levels for detection. Chockalawka Creek was our reference site, if you will remember, and obviously we saw a much higher level of it than at the other sites. There are several reasons. There may be several reasons for this. One could be due to surrounding residences and pastures are having a larger effect than we anticipated. But another reason is it could be the presence of herbicides, which are sprayed along the Talladega National Forest. And